Okay, we're going to learn today about lipids and the cell membrane. The reason for this lecture series on abiogenesis, if it's your first time joining in, this is for you. Dave Farina posted a video entitled Elucidating the Agenda of James Tour, A Defense of Abiogenesis. And I'm James Tour. You can see the description box to his video. I hope you go and watch that so you can see what he said. After watching that video, I was confused about almost every slide and every statement that Dave Farina presented. There were numerous gross scientific inaccuracies in his claims, in my opinion. Every slide of his was wrong. Since others might be likewise confused, I will use the Farina video with timestamps as a launch point for this series of lectures. I'm thankful for Dave. He has his YouTube site, Professor Dave Explains, so this is a shout out for his site. I haven't looked at any of his other videos, only this one, and the only reason, it's not like I happened upon this. There were many people that wrote to me that said, you gotta see this. You gotta see what this guy Dave has said about you and about about the chemistry that you speak about. So it wasn't just about me. It wasn't just about the chemistry, it was a lot about me as well, but I'm not commenting on that. I've got all sorts of problems that Dave doesn't even know about. Dave psychoanalyzed me a lot in that video, and he picked out problems that I have that I didn't even know I had. I know I have a lot of other problems, and so that's not what I'm talking about. It's commendable that he's trying to teach the masses. I don't want any contest with Dave Farina. If you're thinking about a contest and who's winning, I'll just proclaim Dave the winner. How's that? So this is not a contest. I'm just trying to bring clarity because I think that everything that he taught in that video was wrong. Other synthetic chemists, I hope you critique me. Critique me. Point things out. Give me a critique. I want to know that. If I've said things wrong, point that out. If I've said things that are good, you can point those out. Let me know what you're thinking. Do you agree with me on this? Where are we on it? Lipids and phospholipids. A lipid is a compound that shows significant solubility in apolar solvents. That means things not like water. So things more like hexane or gasoline, that would be an apolar solvent. Liquids are defined by behavior rather than by precise structure. They typically contain a polar functional group, but have significant hydrocarbon content. So they have a polar head group, something that's soluble in water, and they're hooked to a tail that generally by itself would never be soluble in water. Phospholipids are lipids that contain a phosphate group. Membrane phospholipids are built on a glycerol scaffold. This is glycerol, this compound right here. It could be called glycerin or glycerol or 1,2,3-trihydroxypropane. Two of the hydroxyl groups of glycerol form esters to fatty acids. Glycerol is achiral, but it bears enantiotopic hydroxymethyl groups. So this is achiral, it's not chiral, but these are enantiotopic hydroxymethyl groups, meaning that if you functionalize one of these, immediately that then becomes a stereogenic center. And so then it would become a chiral molecule. When the two fatty acids are attached, the glycerol component bears a stereogenic center and the molecule is chiral. So as soon as you attach one to one of these hydroxymethyls, you get a chiral compound, and then usually you have both of these, and then this one attaches usually to a phosphate and then an ethanolamine molecule, but we'll see that. The remaining hydroxyl group of the glycerol backbone is often connected to phosphoric acid as a phosphate ester. So you get this phosphate ester forming. So this is your typical lipid with this phosphate group on one end. The phosphate is then connected to a protonated ethanolamine molecule in another phosphate ester linkage. So this is ethanolamine. It'll come in and now you have a homochiral system, and its stereogenic center. And we're keeping in mind, we're just thinking about the simple phospholipids with one stereogenic center. Sphingolipids have 4 possible stereoisomers, and only one exists in nature, the 2s3r. But we'll just keep it simple here. And so this is a typical lipid where you have something like this. Structure of phospholipids: polar head group and a hydrophilic group that interacts favorably with water. A nonpolar tail, a hydrophobic group that is not well solvated by water. So it's got a polar end, nonpolar tail. So the structure of a phospholipid, you'll have these 2 tails hanging down, and then this phospholipid with all its charged and oxygen groups as your polar head group. This is what interacts with the water. This goes down in the lipid bilayer. This is often represented as a sphere with these groups coming out. This is the stick model. This is the space filling model. When membrane phospholipids are added to water, and particularly when they are subjected to shear forces, they undergo a self-assembly to spontaneously form a phospholipid bilayer. If there are no shear forces, the yields are much lower. So really, if you really want to make good vesicles, you got to put these through shear forces or sonication. A monoacyl lipid destabilizes bilayer formation. So this idea that you can have a lot of monoacyl systems in there, it'll destabilize it. They undergo self-assembly spontaneously such that you have polar groups on one side, the nonpolar groups pointing in, and then another side with another layer of these phospholipids where the polar groups are down here, the nonpolar ends are sticking in, and so you have from the top and bottom the nonpolar groups interacting, and then the polar groups outside. One of these is going to be outside to the biological world of an aqueous system. The inside is going to be to the inside of a cell. All right? Cell membrane transport properties. The phospholipid is generally impermeable to ions, so ions don't get through it. Some uncharged molecules like oxygen diffuse readily through the cell membrane. Substrates that are insoluble in hydrocarbons do not pass through the cell membrane. Substrates that are highly soluble in hydrocarbons get stuck in the phospholipid bilayer. Drugs must be carefully designed such that their solubility allows them to pass through the phospholipid bilayer and enter through the phospholipid layer and then enter into the cell. It takes a lot of design to do that. The cell membrane is a large phospholipid vesicle in which there is a phospholipid bilayer. Bacteria can have a double lipid bilayer, and most have a peptidoglycan layer constituting a cell wall rather than merely a lipid bilayer membrane. So actually bacteria, which we say, oh, we didn't make a eukaryotic cell first, it must have been a bacterium. If that's the case, the lipid bilayer is going to have to be a lot more complex because we have actually a cell wall where we have this peptidoglycan layer. But in any case, you have this cell membrane, and you have one side facing outward toward the water, another side facing inward, and there's these embedded proteins in it. These are essential for any cell function. And these embedded proteins have different jobs. And if you look down over the top, so here's the bilayer, there's this protein in there. If you look down over the top, you'll see that this particular protein allows potassium ions in and out. And it has a particular size that allows ions in and out. You have to be able to keep proton gradients. You have to be able to keep the charge in the cell different than the charge outside the system to keep a good proton gradient. And these integral proteins in this bilayer allow certain molecules to get in and certain molecules to get out. They're very selective. If you just have leaky cells, you're going to have dead cells very quickly. Researchers have identified thousands of different lipid structures in cell membranes. They move in swarms through periods of cell function. It has never been shown that a homogeneous lipid bilayer system can operate as a suitable cell membrane. Although all protocells are just homogeneous cell membranes, it's never been shown that something like that could operate as a cell. You have to have different types. That's all we know about in cells today. Lipid bilayers surround subcellular organelles such as nuclei, mitochondria. They all have their own lipid. So you have your outer vesicle, the lipid bilayer, and then inside you have another lipid bilayer around the nucleus, another lipid bilayer around mitochondria, and that's in the eukaryotic cells, which are themselves microsystem assemblies. Each of these has their own lipid composition different from the host vesicle. So each of these has their own lipids that make it up, the lipid composition. Lipid bilayers have a non-symmetric distribution between the inner and outer surfaces. And this has never been shown, it's never been shown that a symmetric composition can operate as a cell membrane. So if this is outside of the cell, this is inside of the cell, you have a lipid bilayer. The constituents of the lipid on this side, on the outside, are different than these lipids on the inside. Every protocell experiment that I've read about, they're all the same. We have no idea if a cell can operate if you have a symmetric lipid bilayer. Protein-lipid complexes are required passive transport sites and active pumps for the passage of ions and molecules through the bilayer membranes, often with high specificity. Lipid bilayers have vast numbers of polycarbohydrate appendages known as glycans. These are essential for cell regulation. Nobody knows how these were made in a prebiotic system. And so to think that you can just take a bunch of salad dressing and get these sorts of assemblies is wrong. And anyway, your salad dressing, all of that came from biological sources. We're dealing with a prebiotic earth here. So in that video that we're talking about the synthesis. It was written, most lipids are easy to synthesize. Okay? Well, then he shows us a triglyceride. So he's showing us groups coming off of this glycerol molecule. But it has now a stereogenic center because R1 and R3 are different. How did that stereogenic center arise in that homochiral molecule if all of these are easy to synthesize? How did that happen? We have no idea. Some have said...
And lipids like triglycerides can be synthesized easily. Any biomolecule is easy to synthesize.
Well, the improper stereoisomer of the lipids are known not to function in cell operation. And so you can see that article. So you have to have the proper stereochemistry there, or else the cells don't function. You can't have this mixture. And we'll talk more about how important chirality is. In that same video, it says,
amphiphilic lipids spontaneously form ordered structures. As we mentioned earlier, this is how soap works. Bilayer formation is a spontaneous process due to the amphiphilic nature of phospholipids, although in prebiotic absence of phospholipids, the first membranes can have been bilayers or even monolayers of fatty acids or other simple lipids that can easily have arisen spontaneously.
Could you please tell me how a fatty acid on early Earth could have formed, where you have methane molecules could have strung themselves together to get a fatty acid and with a carboxyl group at the end? You want to show me the aldol chemistry that's easy to affect so that you can get a fatty acid? I'd love to see that. Fatty acids today are isolated from natural sources. Just even a fatty acid hydrocarbon chain with a carboxyl group at the end. Try to do that on a prebiotic Earth. Some have proposed hydrothermal vents as a route to fatty acids. And there's this paper from McCullum and Seawald, and this is a review article entitled Abiotic Syntheses of Organic Compounds in Deep Sea Hydrothermal Environments. There have been suggestions that the Fischer-Tropsch type syntheses in hydrothermal vents can afford fatty acids, where carbon monoxide or generally one carbon oxygenated compounds can be reduced with hydrogen. The temperature in excess of 150 degrees and high pressures would be necessary. However, the article above states, quote, while hydrocarbons and other organic compounds have been observed in hydrothermal fluids and associated mineral deposits, criteria that can be used to differentiate the abiotic compounds from those derived from other sources have not yet emerged. Experimental studies have shown that light hydrocarbons and more complex compounds can be synthesized under certain hydrothermal conditions, while other experiments conducted under similar conditions have failed to produce organic compounds except for methane. It is not yet clear what factors control whether or not synthesis can proceed, making it difficult at this time to identify with certainty those environments within natural hydrothermal systems where abiotic synthesis might be occurring, unquote. In other words, thinking about just even where a fatty acid comes from before there's been biology around in a prebiotic system is hard. And even if you look at hydrothermal vents today, it's hard to know what is the source of the hydrocarbons that are coming out. Is that really due to a Fischer-Tropsch type reduction occurring? Or is that due to oil products that are under the ground that are of biological origin? It's hard to discern that. And isotopic ratios can help to discern that, but still it's hard. It is possible that fatty acids may have come from Fischer-Tropsch reactions. We can't rule that out. But again, it's not entirely clear, and they certainly don't spontaneously arise. There's a proposed glycoaldehyde route to fatty acids. So some ideas have come out on this. And this was an article from 1991 where this proposal was first given in the Journal of Molecular Evolution by Arthur Weber. While there was a glycoaldehyde route proposed to fatty acids, no such route has ever been demonstrated under prebiotically relevant conditions. So even making fatty acids is hard to think about being done under prebiotic conditions. It's not so easy. Can fatty acids really serve as cellular membranes, as some claim? The first membranes can have been bilayers or even monolayers. Do you know any example, show me any example of a monolayer acting as a cell membrane? This is grabbing things out of the air and proclaiming it from the rooftop. I just thought of something. I think I'll proclaim that with a high degree of confidence. Let's see what the literature says. Here's an article that came out in November 2020 in Nature Chemistry, the top chemistry journal, Nature Chemistry. And here's what Neil Devaraj writes. Quote, all living organisms harness proton gradients across phospholipid membranes to generate energy. Vesicles formed from simpler amphiphiles, such as fatty acids, are unable to maintain proton gradients, suggesting that diacyl phospholipid membranes appear early in the origin of life. Okay, so this origin of life researcher tells us that fatty acids alone cannot, lipids made from fatty acid membranes cannot maintain proton gradients. So all of this nonsense that you can make this from salad dressing is utter nonsense. Cells don't work that way. We are told in the literature that fatty acids can't maintain a proton gradient. Without a proton gradient, you don't have life. So phospholipids must have been around from the beginning. That's what that says. So you got to stop grabbing ideas out of wherever you grab them and apply them here. It can't happen. You can't just proclaim these things. Whoever does that. One cannot suggest the spontaneous synthesis of fatty acids. And even if there were fatty acids, they would not suffice as cell membranes. Furthermore, this is not how soap works, as some have claimed. All right, so here's what Devaraj's article. So he has this whole article on enzyme-free synthesis of natural phospholipids. This is an article from November 2020. This is state of the art in the best chemistry journal. Let's see how he supposes that phospholipids could have been made in a prebiotically relevant synthesis. Well, what he does is he starts with this lysophospholipid, this lysophospholipid LPC right here. It already bears the stereogenic center, and it already bears the phosphate. Where did he get this? Did he make this prebiotically? No. He bought it from Avanti Polar Lipids Incorporated. And I know the owner of that company, Walt Shaw. And they get this from biological sites. And Avanti obtains the homochiral material from a biological source. They put it in a bottle and they sell it. There's no known prebiotic-like route to this homochiral or even racemic compound. Show me the route to even the racemic compound. You got to have a double bond there. How do you get a Z double bond there? The commercial thiolcholine chloride was synthetically coupled with commercial oleic acid from a biological source. Just even oleic acid. This comes from a biological source. This doesn't wander around on a prebiotic earth, and it doesn't form spontaneously, I can tell you that. It was synthetically coupled with commercial oleic acid from a biological source using a commercial carbodiamide, that's a human-made coupling agent, in dichloromethane, a human-made solvent. There's no dichloromethane on an early earth. Nobody suggests that. Under argon, which is an inert gas, to make the thioester 2B. And you see that on the next page. So this he makes by a non-biologically relevant route, and this he buys. And then using an enzyme-free condition, he allows this hydroxyl to attack that carbonyl and to kick that thiol out, and that will then form the phospholipid of interest. He runs this at a pH of 10.6, which is quite high. So he's got it quite basic here. But okay, we'll give him that, and he runs this at 37 degrees, so that'd be a hot day outside. Let's look today at the syntheses of the origin of life, people. Are they prebiotically relevant? We've seen cases before where it was hard to think that they were prebiotically relevant. Let's look at the one today. Remember what I told you. This is what these guys do. This is their little secret. They do relay synthesis over and over again. They take A and they mix it with B, and they make a series of compounds. One of the ones in that series of compounds is the one they want. But they don't use that. They just make that some other synthetic way, or they buy it, and they go on to the next step. So they take this compound, they mix it with C, they get a series of different compounds here, but they don't use that. They take that, they either make it synthetically, or they buy it. So the poor prebiotic earth would be stuck with this mixture. What am I going to do? And the origin of life researcher says, well, that's your problem, not mine, because I can buy it. And they leave the prebiotic earth stuck with that. They don't know what to do with it. They don't have no idea how the prebiotic earth dealt with it and brought that on to the next step with all these other competing things in there. But that's where they leave it, and they do this over and over again. Okay, so here is Devaraj's prebiotic route. And again, I'm not picking on Devaraj here. I have real respect for these folks. I really do. So Devaraj purchased these starting materials for his prebiotically plausible route. He purchased oleic acid. He didn't make it. All right? Choline, this choline thiol chloride. He purchases this, and he couples these under these conditions that he say could be prebiologically relevant. All right, to get this compound. But he already purchased this. He didn't make this by some prebiotic route. And he purchases this compound, and now he couples these two together. So let's see this. Reaction scheme for the dicyano-diamide mediated condensation. That's this compound, this mediated condensation of oleic acid and thiolcholine chloride. This is one of his main points in his paper here, to form the thioester 2B. So this is the reaction scheme. The comparison of extracted ion chromatography corresponding to M/Z 384. That's the mass spectrum positive ion mode for an analytical standard sample of 2B top and 2B generated in the reaction bottom. A turbid dispersion of oleic acid and dicyanamide was prepared. It was turbid because this compound, oleic acid, is not that soluble in water. Was prepared at 0.1 mLs of aqueous hydrochloric acid at pH approximately 1. So you got a pH of 1 here. Remember in the last slide he had a pH of 10.5. Now he's got a pH of 1. It's easy to do that in a lab. You got to ship these things probably a long distance on a prebiotic earth to get this to happen. In one place it's acid, and one place it's strong base. Into this choline thiol chloride is added, and the dispersion was tumbled for 37 degrees overnight. A small volume of the dispersion is analyzed by HPLC mass spec, and compound 2B could be detected. Well, guess what? By this prebiotic route, he doesn't tell us what the yield is. There's no mention of the yield. Now, if I got this wrong, Devaraj or his student can correct me. If it was in that paper, I'm sorry. We'll get that thing corrected. There's no mention of yield or purity in this reaction, only that it could be detected But Devaraj did not use what he made by this supposed prebiotic-like route. He just made it, and then he didn't use it. Why didn't he use it and go on in the next step? Because the yield was probably really bad, and was probably had a lot of other junk in there, and he just detected, yep, it's there. Remember this slide? Yep, it's there. Let's just buy it and go on to the next step. How convenient. How convenient. And that's prebiologically relevant to them. Prebiotically relevant. All reagents below were purchased for the synthetic process. This is how Devaraj made the compound that he really used. He purchased oleic acid, purchases the choline chloride, and he takes 4-dimethylaminopyridine, which is a human-made reagent, and he mixes it with the 3-dimethylaminopropyl N-ethyl carbodiamide hydrochloride. That's this EDC coupling, this EDC compound. And he was added to a solution of oleic acid, which he bought, dichloromethane, which he bought, all human-made material. This is all human-made. This is isolated from natural sources. And thiolcholine chloride, which he buys at minus 78 degrees under argon. Because you're not really going to find that in a prebiotic environment. This is all human-made. And he separates it by column chromatography, and he gets it really pure, something that nature would never be able to do. No idea. But that's nature's problem, not his problem. Devaraj further suggests a prebiotic route to LPC1A. So he further suggests there is a prebiotic route to this. So here's how he made this compound 2B, and then he suggests a way to make the compound 1A. What he suggests is this. He writes this lysophospholipids, such as lysophospholipidic acid, can be obtained by hydrolysis of cyclophosphate amphiphiles, which have been shown to be prebiotically synthesized from glycerol, fatty acid, and diaminophosphate in the presence of imidazole. All right, so what does all that mean? He says, okay, let me tell you how this thing could have been made in a prebiotic sense. How could this thing have been made on a prebiotic earth? Well, it could have been made from this lysophosphatidic acid. And so if you had a lysophosphatidic acid, this is very similar to this cyclic amphiphile. So this hydroxyl just comes in, attacks the phosphorus, out goes water. And I'll give him that. So somehow, if you could make this, then you could have made this. If you could have made this, you could have made this, without any understanding of how he got that stereogenic center. So this is homochiral, what he bought from a natural source, from a company that sells it from a natural source. But he says it could have been made from this. And so he doesn't show us in his paper, but he refers us to another reference. So we look up reference 19 in the Devaraj paper. And when we look it up, we're going to search for this molecule, because that's what he says you can make in a prebiotically relevant manner. So here's reference 19. It's a paper by Mansi. And in this Mansi paper, he really didn't have it. The only thing he made it with, he didn't make it with the C18 chain with a Z double bond. He made it with a C10 chain. He made it with a C9 chain. He made this with these chains, but not with this chain. This chain's very different now. This is going to be a whole lot less soluble. So it wasn't really made in this paper. Something like it was made that wouldn't have formed the same way. So you see, this is a C18 chain oleic acid derived. This is C10 chain, so it's very different in water, and it has no Z-alkene, and it's not homochiral. There's no stereogenic center. This is racemic. If you're going to make this by any prebiotic route, this is racemic. This is not a stereogenic. This is a stereogenic center, but it's racemic. It's not homochiral. It's got both enantiomers here. But he used the homochiral one. How did he set that center in a homochiral sense on a prebiotic earth? Nobody knows, and Mansi doesn't tell us. But Mansi says, well, if you really want to see the synthesis of this, you've got to look up reference 18 in my paper. So Devaraj has us check reference 19 to the Mansi paper. Mansi says, well, if you really want to see that synthesis, you got to go to reference 18. All right, so we go to reference 18 in that paper. So here's reference 18 in the Mansi paper, and we go to this paper by Krishnamurti in Nature Chemistry, and then we go to look up that synthesis, and it's not quite there. He refers us to the supplementary material that is appended to this paper that you find online. Okay. It's 175 pages. 175 pages. Poor early Earth had a lot of chemistry to do just to make this one compound. So he sends you back to this compound. It's not just this one compound. He describes a lot of related compounds to this. So he takes this glycerol, and he's able to make this cyclic compound. But there's no, this is now a stereogenic center, but it's racemic. He has not controlled the homochirality. And he can take these compounds with this DAP compound. He can mix this glycerol and this, he shows it with the C9 compound, not the C18 compound that we really want it to be shown with. And he can get this cyclic phosphate. And where did the researchers get these compounds? They bought them. They didn't get them from a prebiotic earth type method. They bought them. Even though that Sutherland has showed a synthesis for this, nobody's going to use that synthesis. It's too messy. Prebiotic earth has to deal with it. That's prebiotic earth's problem, not these guys' problem. And what if they had to prepare them by a prebiotic-like route? How would they do that? I'm just wondering. How are you going to make a long-chain hydrocarbon by a prebiotic route? Remember, this is before biology. You can't get this from oil, because oil is derived from biological compounds, presumably. Remember this route? We went through this before. Here was the route to that glycerol. So Sutherland has that route to glycerol, which he published formally. We've looked through this in a couple of different videos. Here's what he prepared. Here's what he wanted to get. It's so messy. He puts it on a vacuum pump. He can pump some of these volatile compounds off. If you put it on a good vacuum pump, you pump propane diol off. You see, you don't get it all off, but you can get some of it off. But how do you get a vacuum pump on early Earth? How do you evacuate something? How do you remove the atmosphere from early Earth? Really hard. How do you get a Rayonette reactor, a UV box? How do you get sealed tubes? How do you do degassing and add these different things? It's crazy hard synthesis. That's not prebiotically relevant, in my opinion. And then what you get is this mess, but does he use it? No, he does this. We get the relay synthesis. This is their little secret going all over again. All right, so then he did it with the C9 chain. He did it with a C9 chain, not C18. And it's enantiotopic hydroxymethyls, but he makes this racemic. What happens when the hydrophobic chain is increased from 9 carbons to 18 carbons, like Devaraj would want? Devaraj never mentions. Devaraj never mentions. And then you look up the supplementary material and you look up the synthesis. None of this is easy. A one-pot reaction of nanonoic acid, glycerol, in the presence of DAP and imidazole. And you mix these two together and you add imidazole and about 4 drops of D2O, which is water, but this way you can look at it in the NMR, into an Eppendorf tube, and the resulting mixture was ground with a glass rod into a paste. I'd love to see that paste with not a C9 chain, but a C18 chain, and see how much different that paste would be. The paste reaction was kept at room temperature and ground for 5 minutes every day. The progress of the reaction was monitored by dissolving a small portion of the reaction in 500 microliters of D2O, just the pH, and then he gets the spectrum of this. He doesn't tell you the yield. How convenient. They don't tell you the yield on these things. Remember, we've seen this before, this game that they play. They don't tell you the yield. Unless it's a really good yield. They don't generally tell you the yield. No yield was reported, but you go to look at the spectrum. So he says, go find figure S118 to S123, and that means supplemental page figure 118 or figure 123. So you find figure 121, that's the one we want. So this is the unclean reaction mixture that he got for the C9 compound. Nothing for the C18 mixture, which was ever reported in this paper, as Devaraj would have needed. The C18 would be much less soluble in the aqueous paste. And even this C9 mixture is terribly unclean. How would a prebiotic earth purify this compound to use it? How would it purify it? Look at all this extra stuff. Here's what he wants to get. Here's an authentic sample. And here's what he got. All of these extra peaks are all the extra sludge that from the reagents that he added. All of these extra peaks are all these extra things that he got. All right? So all these extra peaks in here. They made a clean synthetic sample using modern methods, dichloromethane, dimethylaminopyridine, dicyclohexylcarbodiimide, and column chromatography. So this is now what they use. They use this. This is really what they make. It's all relay synthesis the whole way. They leave poor prebiotic earth stuck with a problem that they themselves can't solve. And then you need the preparation of this DAP. Where'd they get DAP? Well, they bought it, or they made it. Well, they make it. So here's how you have to make it. You take phenylphosphorodiamidite. This compound you can't get from a prebiotic earth. You buy it. There's no way that they've shown to get, these aerial rings on a prebiotic earth to be able to do this. And it was suspended in 4 molar sodium hydroxide. I mean, that's really basic, but okay, we'll give them that. And in ethanol. Ethanol is not a prebiotic solvent. Dichloromethane is not a prebiotic solvent. Vacuum for 2 days is not a prebiotic method. So this is not made by a prebiotic route. You'd have to make this by a prebiotic route for this to be relevant. So you look up the synthesis of this. This is in the Watanabe paper in 1986. And Watanabe cites a paper from the late 1950s, and it's not prebiotic by that route either. So this is just, find Waldo. I mean, I'm going to wear you out by sending you to all these references, and then when you go there, it's not prebiotically relevant anyway. So this is the way synthesis is normally done. You take A and B, you get a mixture of compounds, you separate out AB4, and you take that material that you made and you carry it on to the next step. You separate that out, carrying it on to the next step. To do this, high yields at each step are essential to address the mass transport difficulty. If you start with 1 kilogram and obtain 1 gram or 1 milligram, you can't work with that because you don't have enough to carry it on. But somehow prebiotic earth has to deal with that. Isolating the product almost after every step is essential. That's how you really do synthesis. You have to isolate that product. It must be freed of impurities that consume the reagents and result in intractable products. If you don't free it and get clean material to carry on to the next step, what you'll get is intractable materials as you go along. They consume your starting materials. The molecules needed for life require many steps in order to obtain them. Many steps. And you'd have to do purification all along. That's the way synthesis has to be done. But by not disclosing any of that, and by just buying the thing that they've just made, they don't have to deal with the problem that prebiotic earth would do to have higher yields, to have isolation of products and purification, and the many steps that they'd have to go to do this. In this picture, which we've seen before, this is this prebiotic nucleobases, nucleotides, DNA and RNA. This is their presumed prebiotic route where there's no stereochemical control. These reactions that they use that I say are prebiotically relevant, there's no control of absolute stereochemistry and no homochirality. There's little to no control of relative stereochemistry. Only once in a while will they get, I think like in one case they got relative stereochemistry, but not absolute, so it's not homochiral. There's little accounting of yield. They only tell you if the yield is good. There's little accounting of purification. How do you purify this? There's no accounting for mass transport through the sequence. A prebiotic earth would have none of these luxuries. So is it really prebiotically relevant? I don't think so. Origin of life researchers subject a prebiotic earth to constraints of ultra-low yields, racemic mixtures, impure mixtures, and reagent unavailability that the origin of life researchers themselves are unable to solve because they keep purchasing the compound that they want to go on to the next step. They have low yields at most steps, so efficient mass transport would be impossible. Intractable mixtures are obtained with little or no success in isolating them. The molecules needed for life require many steps in order to obtain them, and they would be unattainable by these routes. They'd be unattainable by the routes that are put forward. To those that make inaccurate claims on prebiotic chemistry, they've said things like,
any biomolecule is easy to synthesize.
even the simplest building block, the lipids, are not simple to synthesize. They are not. And especially when you don't have access to the fatty acids. They're not.
And lipids like triglycerides can be synthesized easily.
Wrong, wrong, wrong. You take this from I don't know where and you proclaim it. Why would you do that?
In prebiotic absence of phospholipids, the first membranes can have been bilayers or even monolayers of fatty acids or other simple lipids that can easily have arisen spontaneously.
Nothing arises spontaneously. There's no selectors and there's no spontaneous arising. Nothing arises spontaneously. Chemistry is not like that. The first membranes can have been bilayers or even monolayers of fatty acids or other simple lipids. But Devaraj contests. In the best journal of chemistry, November 2020, he writes, fatty acids are unable to maintain proton gradients. So you're wrong on that. All right, now I'm not addressing that video anymore. I'm going to make an appeal to my colleagues, my synthetic chemistry colleagues who work in the area of origin of life. You have no control of absolute stereochemistry, homochirality, little to no control of relative stereochemistry, little to no accounting of your yields, little to no accounting of purification, no accounting for mass transport through a sequence because you play this relay synthesis game. There is enormous human intervention in every phase of the experiment. Really good synthetic chemists work on this. There's enormous human intervention. Would never be available on an early Earth. You don't make mention of that. A prebiotic Earth would have none of these luxuries. The origin of life researchers, those superb synthetic chemists, are generally silent on these essential synthetic questions. Reading the articles by the origin of life researchers leaves enormous holes which render their work prebiotically irrelevant and misleading the non-synthetic chemist. It is because of the claims that are made in these papers without qualifying all of these problems that people like those who made that video, like Dave Farina who made that video, he's terribly misled by this. And doesn't it bother you to hear the claims that he made? These silly little claims that we showed on the last slide of any biomolecule is easy. Where did he get that idea? From you. That's where he got that idea. And then what he does is he makes videos, and 1 million subscribers hear these videos, and school teachers go into their classrooms and teach it, and homeschoolers learn this. And even professors in the academy are confused by this thing because they too believe a lot of the things that he said. This is not isolated to 6th graders. Don't you feel bad that people think of any biomolecule as easy? Where would they get that? This is not for lipids only. It is for almost all origin of life researcher syntheses. Though this happened in I pulled this all together in the lipids chapter only because the lipids video, because this is the video where I finished the 4th class of the necessary life compounds. This is the problem with it all. So I appeal to my origin of life researcher colleagues. I appeal to you. You got to help me out here in saying, yeah, that there's so many problems here that really when you get to a prebiotic Earth, we're clueless. Because it's not just people like Dave Farina that are misled on this thing. It's famous biologists, like even those at Harvard, that think we're really close to cracking this problem, where we're nowhere close, and you know it. You got to step forward and help me. Reichert, who's stepped forward and published an article and said, you know, you got to have no human intervention, and you got to have, you can't keep using these purified chemicals and claiming that you've got this synthesis worked out. It's folks like that that are beginning to speak the truth, and I really appreciate that. That's going to help the field. Let's work together to get this message out to the world. We have no idea where it came up with, and then maybe, just maybe, some students would come forward, and they'll start thinking about other ways that we could approach this, other ways where we could deal with these mass transport problems, the homochirality problem, that they'd think, way out of the box, not like we're thinking. And they might come up with some solution here, because it must have been formed somehow. So with that, I'm going to wrap up. We still have to talk about a few other topics. Chiral-induced spin selectivity. Is homochirality really necessary for life? And then constructing a cell, all the assembly problems. And then we'll have one global summary. And then hopefully we'll be done. So if you want to hear about where the next video is, you can subscribe right here. And we'll ping you when the next video comes out. It shouldn't take too long. And you'll see that next video. Thank you for joining me. Thanks for joining us. If you want to subscribe, just click right here, subscribe, and we'll give you a shout out when the next video in this series comes out.